The cell cycle is divided into two phases, interphase and the mitotic phase. The mitotic phase consists of mitosis and cytokinesis. Mitosis is the division of genetic material in the nucleus of eukaryotes. Mitosis occurs in somatic cells and during embryonic development. Mitosis is important for living organisms as it is responsible for cell replication, cell growth, and the repair of damaged tissues. The cell cycle starts with interphase, which consists of G1, S, and G2. During G1, the cell grows and carries out protein synthesis. During the S phase, the DNA, which is in the form of chromatin, is duplicated. During G2, the cell grows more in preparation for cell division. During prophase, the nuclear envelope and nucleoli are hydrolyzed. The chromatin condenses to form chromosomes. Each chromosome consists of two identical sister chromatids which are attached at the centromere. The centrosomes migrate to opposite poles of the cell and the mitotic spindle begins to form. During metaphase, the sister chromatids line up on the metaphase plate, which is an imaginary line in the center of the cell. The centrosomes have reached the poles of the cell and they release microtubules which attach to each sister chromatid at their kinetochores.
During anaphase, the microtubule is attached to the centromeres, pulling the sister chromatids to opposite poles of the cell, allowing them to form two identical chromosomes. During telophase, the chromosomes are now gathered at poles of the cell. The nuclei, nuclear membrane, and nucleoli are reappearing. The nuclei are identical to the nucleus of the parent cell. The nuclear membrane surrounds each group of chromosomes. The chromosomes unwind and become chromatids. Cytokinesis is the division of the cytoplasm. In animal cells, a cleavage furrow forms from the membrane squeezing the cell, allowing it to divide into two identical cells. In plant cells, a cell plate, which will become the cell wall, forms, dividing the two daughter cells. Mitosis yields two diploid daughter cells that are genetically identical to the parent cell. The reduction of genetic material occurs through meiosis. Only germ cells, which are present in the gonads of animals and the sex organs of plants and fungi, can carry out meiosis. Meiosis is important because it allows for genetic variation within cells and the production of gametes. During prophase 1, chromosomes condense and form homologous pairs, which are connected by the synaptonemal complex. During the state of synapsis, crossing over occurs, in which genetic material is exchanged, facilitating genetic recombination. During metaphase 1, pairs of homologs line up at the metaphase plate. The chromatids of each homolog attach to the microtubules of opposite poles. This is another mechanism for genetic variation. During anaphase 1, homologs separate and move toward opposite poles, but the sister chromatids remain attached. During telophase 1 and cytokinesis 1, daughter cells begin to separate. In animal cells, the separation occurs through the formation of a cleavage furrow. In plant cells, the separation occurs through the formation of a cell plate. Each daughter cell contains its own set of chromosomes, resulting in two haploid daughter cells.
The next phase of meiosis is meiosis II. This stage involves the separation of sister chromatids. During prophase 2, each chromosome in the cell has two sister chromatids. In addition, the nuclear envelope and the nucleoli are hydrolyzed. Finally, the mitotic spindle forms as centrosomes move to opposite sides of the cell. During metaphase 2, sister chromatids line up on the metaphase plate. Centrosomes release microtubules that attach to the centromeres of the sister chromatids at the kinetochore. During anaphase 2, the microtubules attach to the centromeres of the sister chromatids, pulling them to opposite poles of the cell. This allows them to form two chromosomes. During telophase 2, the chromosomes are at different poles of the cell. The nucleoli and nuclear membrane begin to reform, and the chromosomes unwind to form chromatin. During telophase 2, cytokinesis commences, which yields 4 haploid daughter cells. The result of meiosis is the production of 4 haploid daughter cells that contain one set of chromosomes. The chromosomes exhibit genetic variation from the parent cell.
The difference between meiosis 1 and meiosis 2 is that in meiosis 1, homologous pairs of chromosomes separate, which yields 2 haploid daughter cells, and in meiosis 2, sister chromatids separate, yielding 4 haploid cells.
Mitosis yields 2 diploid daughter cells that are genetically identical to each other and to the parent cell. However, meiosis yields 4 haploid daughter cells that exhibit genetic variation from each other and from the parent cell. This is a result of the crossing over of tetrads in prophase 1. Mitosis has one cell division, while meiosis has two cell divisions, a reduction phase and a division phase. Meiosis II is very similar to mitosis, though. Both of these processes involve the separation of sister chromatids.